Okay, in the past few lectures, what we have been looking at is different aspects of the immune response. So, in today's lecture, what I will be focusing on is to try and get it where all these things come together, which is, in terms of one of the main aims of having the immune system is that it responds to different types of pathogens and different types of tumors. But how does it all come together? Now, in the past few lectures, we have been looking at different aspects of this. Over here in today's and the next one, we will be trying and seeing how the immune system integrates in generating this response. And therefore, coming to the title of this lecture, it is host response mechanisms during infectious diseases. And we will be covering some infectious diseases, but the two main ones that we will be focusing on are the mycobacterial tuberculosis and HIV, because I feel these two represent two ends. One is a bacterial disease and the other is a viral disease. Okay. So, before that we will just discuss some general concepts. Now, what has been happening, and I am sure you understand this, that the way a pathogen causes diseases, there are different types of it, and this involves different interplays of both the pathogen and the host. So, what is happening is there is this constant fight between the pathogen and the host and trying to see who will overcome each other. So, and there are different responses over here. One is you have the humoral and cell-mediated immune response, which is something that I am sure you all have all that several of the lectures have covered over here. Then you have the complement system, and you have also what is important is the coagulation and the fibrin and the clot formation. So, these are all important aspects of it. Now, what is happening is over the years we are trying, we are able to understand better the different strategies that microbes have used in order to in order to subvert the host. Similarly, host mechanisms that are involved in various responses during infection are also coming to light. So, that is what these lectures are there for. Okay. So, this one tells us about the different pathogens that are there. You have viruses, you have bacteria, you have fungi, you have parasites, so on, and they are now coming in touch with the host, and they are trying to see if some of these if they would be able to replicate and use host as a better niche to replicate. So, in order to do that, they would have to infect, they would have to grow within, find out mechanisms by which they can infect the host and increase their cell numbers and disseminate their progeny. So, that is the most important aspect, and the host, of course, cannot be quiet to this, and it will naturally respond, and these are some things that you have talked about. You have different soluble factors, you have enzymes, the complement, the acute phase proteins that are made in the liver, and you have different kinds of cells that are important. You have the neutrophils, the eosinophils, the basophils, the macrophages, and then you also have the cells of the adaptive immune response, which is your B cells and T cells, and there you have the cytotoxic T cells, the different types of helper cells. These make cytokines, and they try and get a better host response. Okay. Now, pathogens are defined as microbes that are capable of causing host damage. When host damage is reached, it beyond a certain point, it manifests itself as a disease, and therefore, the evolution of infectious disease in an individual is a complex interplay between the pathogen and the host. Okay, and what are some of the important general features of immunity to pathogens? One is that the host response is mediated by both the innate and the adaptive or the specific immunity. The innate immune response plays an important role in determining the nature of the specific response, because the initial response is really the key. So, if you have a good initial response and the innate system is able to manage it, then there is perhaps no need to get an adaptive response coming. But however, often because of the replication time or the quick replication of pathogens, they are able to subvert this process, and therefore, you need both innate and adaptive to come in and play. The adaptive is also important not only in generating a specific response, but also in generating a memory response, which may come into play years later after the first time the pathogen has infected the host. Okay. And there are of course different distinct and specialized cells as well as molecules that are involved in this, and this is something that we will be discussing. Now, the other important aspect is not only is the host response important, but sometimes what happens is that the host responds to a pathogen in such a way that it hurts itself. This is a very important aspect, and which is often not so well appreciated, because often the onus is put entirely on the pathogen, that the pathogen is causing response. But sometimes there is pathogen infection, the host tries to take care of it, and sometimes it generates an exaggerated response, and this is which causes organ damage and hurts the host itself. So, upon infection, host sometimes responds, and the response sometimes is a little far greater than what is required, and it causes damage to the host itself, and this is an important aspect. Often necrosis and all that we see is often a result of that. Okay. So, this slide is sort of tells us about the microbes and some of the initial cells that they come in contact with the epithelial cells, and some of these epithelial cells make antimicrobial peptides. Then you have contact with phagocytes, natural killer cells, and these happen early on because they are part of the innate immune response. Subsequently, and you can see there are days listed over here, you have specific responses, you have the B cells and the T cells, and then you have antibodies being produced, you have the effector T cell responses which produce cytokines and which will respond against these pathogens. Okay. So, what are the different types of agents that cause infectious diseases, and this is something that I am sure you are well versed with. You have viruses, you have bacteria, you have parasites, fungi, worms, so on and so forth. Let us discuss a little bit some of this. So, in the viruses there are two main kinds. You have the DNA viruses and the RNA viruses, and some of the well known viruses that I am sure you are sort of aware of are you have the corona viruses which are responsible for your common cold, you have the flaviviruses which are responsible for dengue, then a Japanese encephalitis virus, then you have the retroviruses, the most famous of them being the HIV. There are also several bacterial diseases that I am sure you are familiar with, Treponema pallidum, which is important for in case of syphilis. Then you have Salmonella typhi, which is important in case of typhoid. It is a gram negative organism that causes typhoid, which and is taken in by the gastrointestinal route, and it disseminates from the gut into the different parts of the body. And then you have mycobacterial tuberculosis. Mycobacteria is taken up in terms of aerosols, it is ingested by the lung, and it forms nodules or granulomas in the lung. It generates acute cell mediated host response. I am sure these are ones that you are sort of familiar with. Then you have the fungal diseases. Candida albicans is a primary example. Often Candida albicans, for examples, often affects, or these fungal diseases often affects immunosuppressed individuals or people whose immunity is a little bit lowered, for example aged individuals. Then you have the protozoal, you have Giardia, you have Leishmania, Plasmodium which falciparum, so on, which causes malaria. And you have the worms, worm infections. So, you have filariasis, which is often which goes by the name of elephant foot, and again it is a host response that causes that reaction to these worms. Okay. So, in general, in terms of host responses, the first barrier that the pathogen has to deal with is the body surfaces, and over here you have a natural barrier, plus our body has a lot of commensals, and so therefore, this pathogen has to compete with the commensals that are already present in there in order to get an infection going. And also some of these some of the bacteria that are present in our gut flora or on our body surfaces, these produce antibacterial coalescence which we know which would target other bacteria. Now, in case of for the lung, for example, you have phagocytes in the lung, and and there is a acidic pH on some body surfaces. So, these would certainly hurt the organisms that plan on trying to pathogenize the host. Then there are other mechanisms. For example, our tears contain lysozyme, which specifically targets the cell wall of bacteria. Then you have also secretion of surfactant proteins, for example collectins in the lung, and these are surfactants. So they would sort of they would act as soap, and so they would sort of dissolve the cell wall and membrane of different pathogens. One important strategy, of course, is the production of antimicrobial peptides. In fact, the production of antimicrobial peptides is a very important aspect, and this is often done by cells known as the Paneth cells in the small intestine, and you have a lot of cells in there, a lot of microbes in there, and the antimicrobial peptides is a very important part of that mechanism. Then the other ones are ones that you have sort of heard and have been discussed in these lectures. The complement, for example, is very important. Now over here you can have specific immunity. In some cases the alternate pathway of complement is activated in the absence of specific antibodies, because some of the microbial molecules that are present on microbe surfaces activate or turn on the alternate pathway, and so that would result in activation of the complement and lysis of this. You also have natural killer cells, and these secrete important cytokines like interferon gamma, which activates macrophages, and again this is important. Then you have the adaptive immunity, which of course would kick in a little bit later. You have dendritic cells. These would ingest these microbes, travel to the lymph nodes, where it would initiate specific immune responses, B and as well as T cell immune responses against these invading pathogens. What are some of the key molecules that are involved in the host response? One of the first ones is MHC, and MHC has been very well discussed, but certainly MHC is very important because what they do is they present peptides or pathogen specific peptides onto your T cells, and so there has been association between diseases and the particular MHCs. For example, this particular allele HLA-B53 encodes resistance to malaria strains in Africa. Then you have the HLA-DR2 and the DQB1are, which is associated with pulmonary tuberculosis. In case of HIV, again HLA-B57 B27 are protective alleles, whereas B35 encodes susceptibility for HIV disease progression. So these are cause and effect relationships that have been studied, especially in populations where they look at cohorts and they find which populations or which individuals are susceptible or resistant, and can there be a correlation with the MHC. Perhaps one of the most important features of the ability to get rid of pathogens is the ability of the host to generate neutralizing antibodies. This is the most fundamental feature. In fact, the vaccines that we have today are ones that elicit good robust neutralizing antibodies. What do we mean by neutralizing? That means the antibodies that are produced, they neutralize the microbes. That means they neutralize the bioactivity and action, so that these microbes are no longer able to replicate. So the antibodies can be against particular toxins, or they can be against whole viruses, for example. Smallpox is a good example. So the ability of the smallpox vaccine to eradicate smallpox is because it induced potent neutralizing antibody titers, and such that that smallpox could not recover and has not been able to come back, because the smallpox vaccine strategy have been so successful. So one of the big goals of having a good vaccine is, or one of the big aims is, that it elicits good neutralizing antibodies. This runs into problems, of course, with organisms that generate a T cell response and not that much of a B cell response. So HIV, for example, one of the big problems has been is to generate good and sustained anti-HIV neutralizing antibody titers, and especially if the organism is smart and it mutates very rapidly, then it becomes a problem because it often fools the host, because you can generate a particular antibody response, but by the time you generate it, the organism has changed and mutated into a different form. So then of course it becomes problems. But overall, one of the big successes of vaccines have been or the ones that have been successful are the ones that have been able to elicit a good robust and neutralizing antibody response. The other factors that are involved are the cytokine or the cytokine patterns. So, for example, Th1, Th2 responses. In case of Leishmania infection, for example, black 6, C57 black 6, which is primarily a Th1 producing or produces high amounts of Th1 cytokines and are able to resist Leishmania, whereas Balb C are more susceptible because they tend to be Th2. Now, the ability to produce certain inflammatory cytokines are also important, because IL-12 mice that lack IL-12 or that do not produce interferon gamma are highly susceptible to infection with mycobacteria. So, it tells us the importance about IL-12 and interferon gamma in determining susceptibility to mycobacterial infections. And this is not only true in mice, it is also true in humans, because those who lack expression of these are highly susceptible to infection. And we will be discussing this a little bit even when we come to vaccine, because if you give a live vaccine to patients who lack these molecules, they come down with disease. So, that is again a problem, but it tells you about the important roles of interferon gamma, IL-12 in determining susceptibility. Now, the Th1, Th2 profile, as has been discussed in the previous class, has other important effects also. So, for example, when we have worm infections, for example, the worms produce eggs so that they can disseminate. So, it goes out in the feces and so it disseminates. Now, what the Th1 cytokines do is that they inhibit egg expulsion. So, they form granulomas over here, which causes more host related problems. The eggs get stuck and they result in granuloma formation, it causes problems. The Th2 on the other hand, if the host is more Th2 type, then they would stimulate mucus secretion, there would be changes in the gut epithelium so that the eggs would be excluded. So, you would not realize that this person has a major problem because the eggs are excluded, the host response is somewhat different. So, what this is telling us that the type of cytokines that are produced are important in determining what is the response to these different organisms, and the worms are a very good example of this. Okay, the other place where you see very stark differences between Th1 and Th2 responses are in case of leprosy. So, in cases of leprosy, there are two ends of the pole. One is the tuberculoid form, where the immune response is primarily Th1 and the disease is contained in terms of tubercles, and the other is the lepromatous form, where disease is disseminated. Now, what has been seen is that patients who are primarily in the tuberculoid form show a Th1 response, whereas those who show a more lepromatous form are mainly Th2. Now, this of course, the TT and the LL that are discussed are two extreme forms of the disease, but usually they are the ones are in between. So, you have bottom line tuberculoid or bottom line lepromatous, and so these are in between cases. So, you probably have a mix, but at the two ends are the polar ones, where you have either Th1 or Th2, and that has been shown very nicely in several studies. Now, human T cells from TT patients in fact respond to mycobacterial antigens, but T cells from the lepromatous ones are thought to be anergic. And so, the human T cells are able to respond and they generate this Th1, and whereas the ones from the lepromatous LL ones are not good at, and in fact are thought that they are poor responders to these antigens. Okay, the other cells that play an important role are your natural killer cells, and here natural killer cells are important because in some cases they are important in response to viral infections, and this came out when with the discovery of CMV1. Now CMV1 was a locus which was encoding resistance to a particular virus, and people were not clear as to what the mechanisms were. Subsequent studies showed that CMV1 encodes NK receptor as an activating NK type receptor LY49H, which binds to CMV or a mouse cytomegalovirus virus, and it generates some sort of a response. So what this showed was this interrelationship of NK cells or NK receptors, host encoded NK receptors, in determining susceptibility to viral infections. Now all molecules are not the same. Some molecules can have some sort of a role in some diseases, but not the other, and that is shown in case of CD40, where CD40 mice are very susceptible to leishmania, which means CD40 plays an important role in determining resistance to leishmania, but there does not seem to be any role with mycobacterium, perhaps because there are other molecules that are important and play a role in this case. Now TNF alpha is tumor necrosis factor alpha is clearly an important player in the immune responses, and what has been shown in case of rheumatoid arthritis is that as anti-TNF treatment is given. And what anti-TNF treatment does is it ameliorates the disease symptoms of this autoimmune disease. Now ever since anti-TNF treatment has been going on in the clinics, what people have found is that with the rise in the use of anti-TNF, there is a concomitant rise in the cases of TB. So it clearly shows the role of TNF alpha in determining resistance to mycobacterial diseases. So previously, the moment anti-TNF was given, it helps you against rheumatoid arthritis, but what it also does is that it makes you more susceptible to TB. So if you have been exposed and in previous cases you had enough TNF alpha that was sort of taking care of this, now in the absence of that, now there is the cases of susceptibility rises, and you have more cases coming down with TB. Now also TNF alpha, tumor necrosis factor, it enhances necrosis. So it enhances NF kappa B, which increases. It also activates caspases and results in death. So it also plays some sort of a role in terms of organ damage. Now interleukin 6 is another cytokine which is important, and what has been shown is, and this part was covered in when we discussed the Treg part or regulatory T cell bit, is the role of regulatory T cells is to suppress immune responses. Now if immune responses are constantly suppressed, how do you generate an immune response when a pathogen comes in? So once a pathogen comes in, one of the ways it does through the TLR is to upregulate production of IL-6, and now what IL-6 will do is that it reduces the activity of the Tregs. So the Treg function is suppressed, and you have a more anti-pathogen response that can be initiated. So clearly IL-6 plays an important role in this, especially in initiating the Th1 and the Th17 TN mediated responses. Okay, one important molecule which is important in terms of which plays important roles in infectious diseases is NRAM, especially intracellular bacterial diseases against salmonella, against mycobacterium. And what was found is, and let me just tell you a little bit of a story. When the New World was discovered, New World being the Americas, you had the European population that went in and was taking over land and the people. Now the endogenous people, which is the American Indians, they were not exposed to these the diseases that the Europeans also brought along with them, especially mycobacterial diseases, and large populations of them succumbed to these infections. And it was really in fact a tragic scenario, because you had several people who had never been exposed to this, so their host, they had zero host immunity against these, and they succumbed greatly. Now during this process, what people try to find is what are some genes that are involved in encoding resistance, and using these similar studies people came up both in human as well as in mice on a molecule known as NRAM. And so that is the name of this. NRAM is a natural resistance associated macrophage protein, and it determines susceptibility to intracellular infections as was mentioned. So what NRAM is, it associates with the intracellular, it's present intracellularly, and it is important in transporting metal ions, especially iron, manganese, and so on. So it's about a 56 kDa protein, and it's got a transmembrane predicted 12 transmembrane predicted regions, and associates with the lysosomal membranes. Remember, these parts are important because often intracellular bacteria, they harbor in certain vesicles, and these vesicles are similar to lysosomes, but they actually don't fuse with lysosomes, because if they fuse, then the bacteria would be ingested. So they are similar, and so it's interesting that NRAM is found in these sort of vesicles. Okay, now why are these ions like iron and manganese important in terms of intracellular replication? So it turns out that for bacterial replication they need these ions. So for example, in case of M. tuberculosis, the more iron you give, the more the bacterial growth, because its iron is required for replication. And there are different forms of NRAM. So you have different alleles of NRAM. So you have a NRAM R allele, which is the resistance allele, and this increases macrophage activation, nitric oxide production, and pathogen clearance. Whereas you have the NRAM susceptible allele, which is opposite, and so it induces primarily Th2 responses, which sort of suppress this. So clearly NRAM plays an important role, because especially depending on the allele that is there, it is important in determining susceptibility to infectious diseases, and that is what is sort of shown over here. You have pathogens over here, and that is what I was saying, that the pathogens try and hide away from the lysosomes, and the NRAM is there, and NRAM is probably important in transporting these ions and in determining susceptibility. So if you have NRAM R, then you have a heightened host production against the pathogen, whereas if you are NRAM S, so you know the pathogen has a better chance or better ability to grow and replicate within the host. So very important, and NRAM is a very nice study in this case, and therefore it's important for students to understand the role of NRAM in determining susceptibility to intracellular pathogens. Okay, so we will now discuss some aspects in terms of bacteria. So immunity to bacteria is is often due to antibody production, and this is true especially in case of extracellular bacteria. In case of intracellular bacteria, it gets a little complicated because the bacteria is residing within a cell, and within a cell, how do you get rid of that is much more complicated than extracellular bacteria, which is in a sense more straightforward in that sense. And so if you generate a good antibody response to it, you should be able to manage it. So the extracellular bacteria, they are extracellular because they replicate outside of host cells, and they induce disease by different mechanisms, one of which is they induce inflammation, they produce toxins, by endotoxins, exotoxins. Again, endos are associated within themselves, exotoxins are released outside. So again, a good way to deal with these extracellular bacteria is to generate neutralizing antibodies, as was mentioned. Now one of the apart from the neutralizing antibody response, which would take some time, you have phagocytosis by neutrophils, monocytes, and tissue macrophages, which would be part of the innate response, and which would take care of that. Now the activation of the complement system is again important. Now the humoral immune response, which is, we said, the antibody mediated response or the neutralizing antibody response was an important one. What has been seen is that the strong IgM response caused by polysaccharides, and then antibodies IgM and IgG, these stimulate different types of mechanisms by which these antibodies are generated, and they play an important role. Now, there are different types of effector mechanisms. The Ig antibodies opsonize bacteria. So, these would bind to bacteria, they would opsonize it and they would enhance phagocytosis. Now you have macrophages and neutrophils that would be able to ingest this much better. The antibodies may neutralize bacterial toxins, that would be a more straightforward way, and IgM and IgG can activate the complement system. Now one of the host responses to extracellular bacteria are, again, if you have too much of it, it results in inflammation and septic shock. And now this again, septic shock was something that was discussed in great detail in my class on innate immunity, and I would urge students to look that up, because that's really an important part, because especially in hospital infections, a large majority of hospital cases where you go in with a particular case history, but you have some disease that is often due to septic shock. Often also hospital cases are associated with infections with the drug resistance bacteria. So, septic shock is something that is important. Students need to look this up, and I will not discuss it over here because it's been discussed in the class on innate immunity. Okay, so this sort of gives us a little bit of a review. You have microbes, and you have your B cells, and you have an antibody response. You have neutralization of your toxins, of the microbial toxins. You have opsonization and Fc mediated phagocytosis. So they are sort of taken up. You have bacterial lysis because you have activation of the complement system. You have inflammation and then phagocytosis of the complement coded bacteria. Okay, now as previously mentioned, intracellular bacteria is a little bit more problematic, primarily because they have the ability to survive and replicate within phagocytes where they are inaccessible to circulating antibodies. So, this is a little bit of a problem, and so elimination of intracellular bacteria requires immune responses that are very different from that for extracellular bacteria, because in that sense extracellular bacteria are more straightforward. Okay, so what has been seen are intracellular bacteria. They grow within phagocytes, okay, and so they are able to resist lysis within the phagocytic machinery, which is what I said. They do not fuse with lysosomes, they stay alive and replicate in vesicles other than that. These are also activate, so IL-12 for example, they activate natural killer cells, and natural killer cells can stimulate by production of cytokines, and for example IL-12. Now, the way to get rid of intracellular bacteria, there will be two main types. One is you have production of cytokines like interferon gamma, which would activate the macrophages or phagocytes, and they would then the heightened activation would result in increased production of reactive oxygen and nitrogen intermediates, increased production of cytokines, and that would sort of take care of the bacteria. The other way is to have lysis of the infected cells with cytotoxic T lymphocytes. So, in which case you would generate a T cell response, and a part of the T cell response would take care of this bacteria. In infections with intracellular bacteria, you have tissue damage, and the macrophages often accumulate over here because they are trying to contain the disease, and this results in granuloma formation, and this is something which we will study a little bit in greater detail when we discuss Mycobacterium tuberculosis infections. Okay, so let me just briefly discuss one intracellular mycobacterial infection before we go on to Mycobacterium tuberculosis. Now, this is the case of Salmonella. In case of Salmonella typhi is a gram negative bacteria which is ingested by through the oral route, and so it enters the gastrointestinal system, and from there it is disseminated systemically to the body. That means from the intestines the bacteria enter the body through the intestines. So they are able to colonize through the intestinal route. And what it results in fever, it results in a manifestation known as typhoid. So what are the host responses over here, and this slide sort of tells you about the different phases in case of Salmonella infection. So you have, this is the initial phase, and then you have the phase 2, which is the peak phase, and then by phase 3 they have sort of they have been contained because the host has sort of taken charge and has contained. Now once it has come here, there can be a relapse, they can enter into a carrier state, or you have the antigen specific immunity coming down, as a result of which it comes down. So that is what is shown over here. So in fact over here it has been shown as phase 3, but in case sometimes it can even go up if the response from the host is not good. But what are the host responses, and again it's shown kinetically, so you can see that. So the first response is obviously the innate resistance. So you have end ramp, which is important, oxidative burst, complement, sort of which are important in the first phase. Then you have the adaptive responses, and here you have the interferon gamma, the IL-12, the granuloma formation, the production of nitrogen intermediates, the complement, so on. And then subsequently at a later point you have antigen specific immunity. So for example, you have the CD, you have the T cell response, you have the CD28, which is the co-stimulatory receptor, the MHC class 2 response, and so that is what is shown over here. So you can see the initial one is the innate, and the later one is the antigen specific response, and right over here you have sort of the adaptive form, where you have interferon gamma, the TLRs and the complement that sort of play an important role over here. But in terms of bacterial diseases, the one that we will be studying in slightly greater detail is tuberculosis. Now tuberculosis, as you are well aware, is caused by Mycobacterium tuberculosis. It is an acid fast bacilli. What do we mean by an acid fast bacilli? So an acid fast one is one that is resistant to counterstaining with acid alcohol. And why is it resistant to counterstaining? Is because the mycolic acid that is present gives it this ability. Most other bacteria are can be easily decolorized and stained with a counter stain, but not mycobacteria, and that is because of its mycolic cell wall nature, and hence it is called an acid fast. So students who are not aware of it should look up and try and understand this acid fast staining procedure, which is important. So it's a wasting illness. It's with fever, cough, and the lung tissue gets affected. In the older days, what people would do is to ask people with TB to go to a hill station and lead a calm, stress-free lives, and try and recover from the disease, because I think stress does not help the host response. In fact, it hurts it. So maybe a calmer lifestyle helps the host in sort of recovering back from TB. Now, TB also occurs when immunity goes down. So again, with increase in the HIV cases, what has been found, one of the common manifestations have been TB. So usually even the ones that don't cause these problems are under normal circumstances, when a host is just about is okay, and there is if the host immunity is lowered, then you have these cases that show up. One of the problems in recent times has been that there are virulent forms of newer and virulent forms of TB. So for example, Mycobacterium tuberculosis strain W, which cause various forms of this disease, and the organism has also changed over. I mean, so you can form the traditional form of TB, but it also infects other organs, and it has newer manifestations of the disease. So tuberculosis is a disease that one has to be very careful about. Now, one of the there are two main hallmarks about TB. One is the tuberculin reaction, which is something we will discuss, and the second is formation of granuloma. Now, in case of tuberculin, now tuberculin is actually the secreted antigens or secreted proteins of M. tuberculosis. So, if these are injected within the skin, then the host, there is a very quick response by the host against it, because what you are doing is generating a delayed type hypersensitivity response, and this can be measured, and this is known as a tuberculin reaction. So, those people who have been exposed to tuberculosis often generate a good tuberculin reaction. And what is interesting is is that people who were tuberculin positive initially and subsequently develop TB, they become tuberculin negative. They become tuberculin negative because their host immunity has gone down, and the bacteria has sort of won. And once antibiotics are given or anti-tubercular treatment is given, the dots and so on, and as their immunity against TB increases, you see the tuberculin reaction coming back on. So, it is a very interesting scenario, and it is something that the student should be aware about. Tuberculin is a very important aspect of study here. The other is the granuloma formation. Now, what is granuloma? So, you see the organism infects us primarily in the lungs, and then it forms a granuloma. So, what granuloma is, because the host is trying to contain the disease, what it does, it forms, so initially you have macrophages that try and ingest it. The macrophage area is often surrounded by T cells. So, right in the middle you have a necrotic patch, and this is because you have dead bacteria, dead cells over here. So, following the central necrotic patch, surrounding ones are the macrophages, which are secreting interferon gamma and trying to contain this disease, and on the outer rings you have your lymphocytes over here. So, the granuloma is a particularly interesting, and it can be seen very clearly. So, this is clearly seen in case of monkeys and in humans. And so, this sort of figure is to give you an example of that. So, this is the central necrotic part, where you see a lot of necrosis and the clearing, and you have the macrophage and cells around over here, and then you have the lymphocytes, which are shown over here. At the outer periphery are the ones where the lymphocytes are, and especially your T cells, which are sort of responding to the gamma, and they are also trying to kill. So, the macrophages and the T cells form this, and that gives rise to what is a granuloma formation. Now, in terms of intracellular growth of M. tuberculosis, as was mentioned, M. tuberculosis enters via the lung. In terms of aerosols, you sort of inhale it, so they are entered the alveolar macrophages via mannose receptors, FC receptors, complement receptors. So, that is how it enters. Now, what is shown is the vesicles, they stay within vesicles, and these vesicles do not fuse with lysosomes. So, in case if they were to fuse, then the M. tuberculosis would be sort of digested by lysosomal enzymes. However, so it is in interest of the bacteria or my M. tuberculosis over here not to fuse with lysosomes. So, M. tuberculosis resides and divides in resting macrophages, not in activated macrophages, but in resting macrophages. Now, how does it do that? What it does is M. tuberculosis recruits a host protein known as coronin. Previously, it was known as TACO, tryptophan aspartate containing coat protein, and which prevents these vesicles from containing MTB from fusing. So, what Mycobacterium has done, it's got very smart. So, it recruits a host protein so that and which enables it not to fuse with lysosomes, and that again is very much in the interest of the bacterium. Now, coronin is associated with actin, and in mice lacking coronin there is greater fusion of M. tuberculosis with lysosomes, and which is the important part, and which makes the important part. So, this is a very interesting. Now, I talked about M. tuberculosis and different host responses. One was the end ramp, where end ramp is important in determining host susceptibility to different intracellular infections. Coronin is a very important one, because in terms of determining in the way the bacteria has got hold of a host protein and tried to figure out how to escape this this fusion. Now, what about some other host responses? Now, what has been and this is something that has been discussed previously, is that interferon gamma and IL-12 are important in determining susceptibility to mycobacteria. This we have covered previously, because these molecules are important in that sense. Now, we also talked about the P47 FOX, which is a component of the NADPH oxidase, and NOS2, which is important in production of nitric oxide synthase, which will result in production of nitric oxide. So, what has been shown is that reactive oxygen intermediates play a contributory role, but NOS2 plays an essential role in the later part of the infection. Now, some years back, a very interesting experiment was done where different knockout mice were tested for their ability to check for susceptibility to mycobacteria. What was shown is the beta 2 knockout mice are highly susceptible, followed by the TAP1 knockout. Now, beta 2 microglobulin, as you can understand, is a component of the MHC class 1. It's important in that sense, and TAP1 is a transport associated. So, both these two were highly susceptible, which tells us that beta 2 microglobulin and TAP1 play an important role. What was interesting is the CD8 mice were then they were followed then by CD8 and then subsequently by perforin. Now, perforin is important in terms of killing, but you can see that the perforin is not playing a very important role, or certainly, or put it other way, beta 2 microglobulin is playing a more important role than perforin. Now, and then CD1 was similar to wild type. Now, CD1 is a MHC class 1 like molecule, which it belongs to the non-classical MHC, which is important in presenting lipid molecules to your T cells, and especially a type of T cells known as the natural T cells or the NK T cells. Now, NK T cells, it's quite obvious from here that the NK T cells are not playing much of a role, but beta 2 microglobulin is playing the most important, and what is important to see is the beta 2 was more important than the CD8s. Now, remember, in the beta 2 knockouts there are no CD8s, but so clearly beta 2 has a role other than just strictly the CD8 part. So, this was a very important part, and you can see the killing in terms of perforin were not playing an important role. So, this I thought was a very important aspect. It tells you about the complexity of the immune response and how, if you change one parameter, there are other parameters that also get changed, and which is important in determining the overall response of the host towards a microorganism. So, a very interesting study by Barry Bloom's group, which was published in PNAS, and I would suggest students to take a look at that to try and understand this aspect a little bit in greater detail. Okay. Now, there have been other molecules that are also important in playing a role in TB infection. So, TNF is clearly important. Now, we had talked about the anti-TNF a few slides back, and how with the use of anti-TNF for rheumatoid arthritis, there is a rise in TB cases. Now, another molecule, which is a lymphotoxin, is also important because it is important in recruiting lymphocytes to the sites of infection in lung. CD4 plays a very important role because of the production of interferon gamma. Now, there are other molecules. So, IL-1 receptor antagonist is important, vitamin D3 vitamin D receptor. In fact, there are some studies which show that if you take in a lot of vitamin D, it helps you with better immunity against tuberculosis. And then mannose binding protein MBP. MBP is important in terms of opsonization. Again, an important part that was covered in our lectures on in innate immunity. Please take a look at it, and its MBP plays a role against infection in tuberculosis. Let us talk a little bit about BCG vaccination. The moment BCG is a good pediatric vaccine. against M. tuberculosis. So, that means that when kids are born, they often, the first vaccine that they get is BCG. What BCG does is that it protects young kids with vaccination. However, the efficacy of BCG vaccine with respect to adults is controversial. There are two aspects that I would like to cover in this slide. One is, while BCG is important and clearly it is a good pediatric vaccine, it enhances Th1 responses, and this enhanced Th1 responses has been shown to reduce cases of allergy in Japanese in a study with Japanese children. And why is that? Because there is a common perception that people who live in cleaner environments, they suffer more from allergies because they are primarily more Th2 like. So, there is a lot of secretion and a lot of allergies because they primarily are of Th2, and so if you give BCG vaccination and you are trying to modulate their immune response to a more Th1. So, I think that makes an important that changes their immune response parameters, and so it becomes a little bit different. The other case is that some kids who are given BCG vaccination, they come down with the disease, and that is because if they lack a proper immune system in terms of functional STAT1, which is important in interferon responses, interferon gamma or its receptor, IL-12 or its receptor. In fact, all these three, STAT1, IFN, IL-12 play a very important role in host resistance to infections. And so, if you do not have it, so the kids who do not have it are highly susceptible to BCG vaccination in that sense. Okay. We will cover the fungal infections a little bit, because in the next class I will be covering mainly viruses and parasitic diseases. So, over here fungal infections, as was previously mentioned, they are primarily caused in individuals where they are either immunosuppressed because of treatment or due to age, because their immune system has been compromised. In case of Aspergillus, these spores are sort of ingested, the conidia are ingested, and these sort of attack the host cells, and they try and germinate. One of the main reasons, one of the main mechanisms by which this is the host response does is that you have neutrophils are the ones that are supposed to attack the hyphae and sort of kill these this sort of an infection. You also have dendritic cells which ingest the conidia hyphae, and they try and maintain this infection. However, the infection that is perhaps well studied or is the one caused by Candida albicans, and you have mucocutaneous, which means it is localized, it probably isn't skin related or present in the ear, to a systemic, and systemic, if it becomes systemic, then of course it is a life threatening infection. And again, what you have is an immune response involving neutrophils, and you have T helper cells which play an important role. Often what happens is that what is seen is these yeast cells, Candida albicans can switch from a yeast state to a pseudohyphal state, and you can see this in mice. In fact, both yeast and hyphae translocate across the gastrointestinal tract and may gain access to blood vessels. Now, the type of responses that are seen, again you have opsonizing IgM responses, you have the gut dendritic cells, and you have T cells that play important role with the cytokines that they produce. Okay, one of the important ways by which this happens is you generate a good response using your neutrophils and macrophages, and you have a good T cell response primarily using your Th1, Th2 responses that are in place. So, neutrophils, as I said, play an important role. They phagocytose fungi for intracellular killing. and they are present in macrophages. Macrophages also generate cytokines, boost up the host defense, and so you have cytokines that are being produced, and that would do it. So what I will do over here is to briefly summarize the talk today. So initially what we had done, we had to cover different agents that cause disease. You had viral, bacterial, fungal, so on, and some of the viral ones, some of the recent ones that are important to cause cold, HIV, these are things that will be discussed, and this class has discussed some aspects of salmonella and primarily mycobacterial diseases. What are the general mechanisms involved in the host response? General mechanisms involve the body surfaces, you have dendritic cells, you have the Th1, Th2 responses, and the Th17 responses. Now, in some cases Th17 plays a very important role, especially in case of fungal infections, as has been seen lately. In fact, as you probably know and has been discussed, is that the interferon and IL-17 are sort of inversely related to each other, and so even in the absence or lowered amounts of interferon gamma, you have very good Th17 responses. There are some aspects that I would like to emphasize. One is the use of immunomodulatory roles. So, due to rheumatoid arthritis, people started giving anti-TNF, but this while it has helped ameliorate rheumatoid arthritis, it has resulted in increase in cases of TB. So, you can see how tickling one aspect of the immune system affects some others. So, it's in a dynamic flux, and so if you change one parameter, several other things get changed. The other was the use of BCG vaccination and atopy. So, use of BCG vaccination increases increases our Th1 response, and therefore and consequently reduces atopy or our ability to develop allergic reactions. The other fascinating story is that of NRAM, especially the role of NRAM, which is involved in transporting iron, manganese, and its ability to determine susceptibility to intracellular infections depending on the allele that is expressed, the NRAMP R, which is codes for resistance, or NRAMP S, and I really think NRAMP is really a fascinating story. We also discussed mycobacterial infections, the granuloma formation, tuberculin, very important aspects for students. You must pay close attention to these. What is tuberculin? What is granuloma? How are these sort of related to each other? Remember, tuberculin, as a person develops TB, they become from tuberculin positive, they become tuberculin negative. As they start recovering, they regain tuberculin reactivity. So, very important aspect. We also discussed the role of coronin during mycobacterial infections. Remember, mycobacteria enter from the lung, and they play an important role. Finally, we discussed some aspects of fungal infections. Fungal infections, as was mentioned, they occur mainly in immunosuppressed individuals, and Candida albicans is an important aspect. The main roles are with neutrophils, and macrophages usually sort of would tend to take care, and then you have role of the T helper cytokines in helping boost up immunity. So, overall, what this lecture has tried to cover is give general ideas about the host response to infectious diseases, and then we studied tuberculosis as a paradigm of an intracellular infection. In the next class, what we will be discussing are mainly viral diseases. We will be discussing a little bit in depth about HIV, and we will discuss other diseases. More importantly, we will be discussing emerging diseases. Thank you.